Welcome to the Women Documenting the Floras of Texas and Oklahoma, a program out of the Armchair Botany program here at the Phyllocology Herbarium at the Botanical Research Institute of Texas in the Fort Worth Botanic Garden. We'd like to welcome you all to the event and let you know that this is approximately a 55-minute presentation by multiple individuals, and it will be followed by a guided transcription blitz led by Ashley Bordelon for those of you that may want to engage with these specimens further. You'll all be muted to begin, so please do submit your questions via the Zoom chat. We will have a Q&A session after all our presenters have presented, but I don't want you to lose your questions, so submit them in the chat and we'll scroll back through it and try to find those questions to make sure we address them. If we don't, please ask them again. They are important to us and we will save the chat. So please do turn off your videos until we get to the Q&A session. And when we do, at that point, if you get disconnected or have connectivity issues, turn off your video. And then access for this recording will be posted online. So please go back to the event page and you will see it listed there. Otherwise, you'll see it at brit.org/WeDigBio. And then lastly, and perhaps most importantly, please do be positive and respectful in your communications. We're all of us here to learn together, and our community is improved for having you be part of that conversation. So today's session, there will be 5 of us that will be presenting to you today. These are staff and volunteers that hail from many diverse backgrounds. And you'll hear Ashley Bordelon, our collections manager, talk about Constance Taylor. You'll hear Sue Frary interpreted and presented by Kimberly Sasson talking about Agnes Chase. You'll hear Alan Lusk, Texas master naturalist, talking about Shirley Lusk, somebody very important to him. Myself, Tiana Raymond, the herbarium director, I will be talking about Gaeta Agilusk. And then last but not least, we will have Nat Rodriguez. They will be talking about Ines Mejia. And then at the end of all those presentations, we'll have our Q&A, and then we'll proceed to a guided transcription blitz for those of you that would like to continue with us. But a quick note for all of you that may be interested, our event today is a kickoff. It's part of regular programming we do, but it's a very special day because today marks the first of a 4-day period called We Dig Bio. This happens twice a year, and it's a global campaign to digitize natural history collections, to take these very physical collections and put digital surrogates of them online. And there are many events at many institutions around the world. So please do consider following up with some of these. There's going to be some other really great presentations in this 4-day period and other great programs that could use some assistance. All right, jumping into a bit of the introduction for today's talk. So as we're understanding what our floras look like in our 2-state region, in particular in Texas and Oklahoma, we look to herbarium specimens. So on this screen right now that you're seeing, you'll see a specimen on the bottom left, on the far left of the screen, and then a zoom in of the label. Each of these specimens is a testament to the existence of that plant species in space and time. And if you look at these labels, there's even more information that can be gleaned from them. Currently, if you go online to the Global Biodiversity Information Facility, there are about almost 1.5 million records for plants observed in our 2-state region. If you look at which one of these are preserved specimens, that number drops drastically down to 254,000, some odd thousand. That part of the reason is that specimens are not yet digitized. Specimens haven't yet been linked and shared to this global aggregator, which is GBIF. And so I'd like to acknowledge that today's presentation is made possible because of volunteers, but also because of funding from the National Science Foundation. The National Science Foundation has funded the digitization of specimens from our 2-state region as part of a much larger project that I'm happy to share more information with offline. So documentation of our collectors, if you look at those 254,000 or some odd records, or you extrapolate out to the 1.9 million that we believe have been collected in Texas and Oklahoma, I'm talking about physical vouchers here. You know, how many collectors have contributed to that knowledge, to that specimen base? Who are they? How many variants, under what names do they go by? So these hundreds of collectors, it's hard to have more information about them than is on the labels. And we have a responsibility to learn more about them. How many of these are women? How many of these collectors are peoples, the First Nations, first peoples of this nation or others? So there's a lot of stories yet to be uncovered and people's contributions to be pulled out, whether it's 30 specimens or it's 30,000 specimens. If you're looking for information on collectors online, one of the best places to pop in to learn the nitty gritty is the Harvard University Herbaria databases. There's a botanist search. You're not seeing the window in here, but there's a botanist search window that you can search for botanists. And I've pulled up a search for Eula Whitehouse in here. And so you can see some of the basics about Eula Whitehouse, where she collected and deposited, when she collected, when she was born, and when she died. So it's an extremely useful database, but we find that so many of our collectors that may have collected fewer specimens aren't in that database. And then we pop over to the work at UNC Chapel Hill Herbarium. And I particularly enjoy what many of those contributors have done in putting together a list of the collectors from their herbarium. And you can see that the way that these collectors are introduced and described is much more in depth and provides a different level, different sorts of information than the Harvard Botanist database does. And so my next slide is talking about a slightly newer endeavor called Bionomia. This is an online effort to identify these collectors, these scientists, these contributors, identify them in a way that that their specimens can be claimed and they can be acknowledged for the work they've done. So what you'll hear about today in presentations from our staff and volunteers are sort of our first forays into talking about the women whose collections we've connected with. But we hope that this is just the very beginning, and we hope that you will, if you have stories, comments, corrections, we hope you'll write to us. And there's our email address, herbarium@brit.org, that you can send those comments to if not in the chat. And let us know, because we are committed to seeing this through and finding a better way to represent our collectors online in a way that other people can access them. So with that, I'd like to introduce Ashley, who will be talking about Dr. Constance Taylor.
Thanks, Tiana. Yeah, so like Tiana said, with this National Science Foundation grant, not only are we looking at Texas, but we're also looking at Oklahoma. And a lot of our Oklahoma collections have the names John and Connie Taylor on them. So we wanted to look into both of them as a team, but also Connie Taylor specifically. So full name, Constance Elaine Southern Taylor. She was born in Washington, D.C. in 1937. And at some point in her young life, her family moved all the way to Oklahoma. A small town in Oklahoma. I think it was Ardmore, Oklahoma, if that's familiar to anybody. She went to the University of Oklahoma and got her Bachelor of Science in 1959, stayed and got her master's in 1961, and continued on to get her PhD in 1975. In college, she met her husband, John Taylor, and they were quite the team. And once she finished her master's, they actually moved down to Durant, Oklahoma, where the Southeastern Oklahoma State University is. And you'll see kind of on that map, it's a little bit past the Texas-Oklahoma border. And I believe there's a park out there as well. So a good location for her to collect, and the herbarium so close. And you can go next, Tiana. So for her PhD, she worked with Dr. George J. Goodman. We actually have a lot of his collections as well. He was her advisor. And they were working on apparently one of the most taxonomically difficult groups in the Asteraceae family, the goldenrods. So this includes Euthamia and Solidago. So some of those weird, funky goldenrods that have a lot of issues. So she tackled that. And I love that in her acknowledgments for her PhD dissertation, she said that her kiddos had become good Euthymia and Solidago spotters and who have spread many blotters out in the sun to dry. So I love that we have some instances where collectors, whole family gets involved, kids and spouses and all of that. And you can go next, Tiana. So in 2001, we actually absorbed the herbarium that had the Southeastern Oklahoma University collection. So John and Connie Taylor started that collection when they had moved to Durant, and I believe in 1969. So she was still doing her PhD. And it had a few other collectors, but they kind of had the bulk of that collection. It was pretty much their collection, the John and Connie Taylor collection. So in 2001, that came all the way to the Britt Phyllocology Herbarium, and just taking a look at what is digitized in our collections that I could search. Just in the Britt collection, we have 8,236 collections of theirs. But across all herbaria, I saw over 17,000 collections. However, this was mostly in the Britt collection. What we're seeing are Texas and Oklahoma collections, because that's been our focus to digitize. But we know that they were avid travelers and adventurers. They basically vacationed to places so they could collect in these places. So not only in the US, but they even went down to Costa Rica and up into Canada too, I believe. So they kind of went all over collecting. And I think some of their highest collection numbers may have gotten into the 30,000s or the 40,000s. And just looking at these two screenshots, I wanted to show that they really were a team. If you know anything about collections, you know that you start with a collection number one and you go from there, each collection getting an individual number, increasing as you go. They collected as a team. So whether John was technically listed first, or maybe it was Connie listed first, whether they collected together or separately with other people, they kept those numbers in sequential order as the years went on. And you can go next, Tiana. And these, this is just a snippet. I mean, they both contributed so much to the flora of Oklahoma and beyond. So these are just a few snippets of a few of her contributions. And really, I mean, like I said, they were really such a team doing this together. On the right-hand side, you'll see there's two species named for them, Castanea tayloriorum and then Elaphoglossum tayloranum. These are both Costa Rica specimens. And then also, what else? There was a documented list of Oklahoma flora that was published by Waterfall in 1969. And since then, I think maybe in the early 2000s, they had added over 300 species to that list for the flora. So they really contributed a lot. And I know they were known for specifically making it a point to not only collect everything on that list of flora of Oklahoma, but to repeat and go back to these locations and check up with these species to see if these populations are still where they were previously. They both published in our very own CIDA. I think that publication was in maybe the early 90s. And I won't take up too much more time, but she was a part of a number of organizations from American Association of University Women, the Oklahoma Ornithological Society, the Oklahoma Native Plant Society. She was a president for a year, and she got numerous awards from many different institutions and societies. So she recently passed away this past year, I believe in August. So really, I mean, she will be remembered by so many people who either knew her or didn't, just based on the work that she's contributed in Oklahoma and even in Texas and beyond too. So that is the end of Connie Taylor, and I'm going to pass it off to Kimberly now to talk about our next botanist.
Thank you, Ashley. I am here to present about Mary Agnes Chase. I'm presenting about her on behalf of Sue Frary, who is a Texas Master Naturalist with the Indian Trail Chapter, and also on the steering committee of the Texas Regional Consortia of Herbaria. Mary Agnes Chase was born in 1869. As was common, her formal education ended with elementary school. She married in 1888 to William Ingram Chase, who passed away a year later, and she never remarried. She kept his name. At the age of 24, a trip to the plant exhibits at the Chicago World's Fair in 1893 really ignited her interest in plants. And 5 years later, in 1898, while she was collecting specimens in a swampy area near Chicago, she met a botanist who encouraged her enthusiasm and taught her to use a microscope to illustrate new species of mosses that he had discovered. She became so skilled as an illustrator that she later worked at the Field Museum in Chicago. In 1905, her illustrations caught the attention of Albert Spear Hitchcock at the U.S. Department of Agriculture in Washington, and he hired her to do his illustrations. Agnes was a self-taught botanist who so impressed Hitchcock that they became collaborators on grasses. You'll see both of their names on many vouchers as collectors as well as determiners. In 1911, there was a big botany research symposium in Panama where all well-known scientists were invited, including Hitchcock, but not Agnes Chase. The Smithsonian officials organizing it were very clear that they only wanted real scientists on the trip. She must have been really annoyed because she funded her own field work in Brazil shortly thereafter, discovering many new grass species. Chase often funded her own field work, although the specimens she documented and collected became the property of the National Herbarium in Washington. Some of her collections can be found in herbaria worldwide, but primarily there in Washington. According to Torch records, Agnes collected grasses in West Texas and along the Gulf Coast in the summer of 1912. A lot of the grass vouchers from the early to mid 1900s in Texas have determinations by her. By the 1920s, she was recognized with Hitchcock as two of the world's primary authorities on grasses. They collaborated on countless collections and determinations from all over the world. Agnes was also an active suffragette, in fact, so active that she was actually jailed in Washington for participating in a demonstration outside the White House. When officials at the USDA tried to fire her for her activism, Hitchcock told them that he could not do his work without her, and they relented. Without Hitchcock's intervention, we might never have had the work that Chase has given to botany. Her teaching book, First Book of Grasses, which I happen to have a copy of here from our herbarium, published in 1922, is still used for beginning botanists, though it's a pretty hard read if your interest in grasses is only casual, but she intended it for the serious but amateur student. It's available on Amazon and updated recently by Richard Pohl. In 1936, Agnes was promoted to the role of senior botanist and became responsible for the USDA's entire systematic agrostology department, the grasses department. She retired in 1939, but maintained her role as custodian of the grasses at the U.S. National Herbarium for the remainder of her life. In 1958, Chase received her only college degree, an honorary Doctor of Science awarded by the University of Illinois, and became the 8th honorary fellow of the Smithsonian Institution. And in 1961, 2 years before she passed away, she became a fellow at the Linnean Society of London. Thanks for listening about Agnes. Next up is Alan Lusk.
Hello, everyone. It's my privilege and honor to be able to talk about my mother. I'm a master naturalist and also a member of the Native Plant Society of Texas. Just got back from their symposium down in Alpine. My mother was born back in 1924 and passed away about 5 years ago now. She was actually born in El Paso. My granddad happened to have a dairy there back in right after he got out of college. They, after a couple of years, moved to Hermosa Farms in Dallas in 1928. And the picture on the right is a picture of my mother working on the farm, raising chickens, and I understand she also raised turkeys as well. Kind of unusual, she received a Bachelor's of Science degree from the University of Oklahoma in 46. She was actually working in the labs after she got her degree down at Texas A&M, where she met my father, Dr. C.F. Lusk, and they got married in 1950. My dad was working as an entomologist there, checking cotton fields to see what they needed to spray. You know, from the 50s up through probably the early 80s, she spent most of the time raising us 5 kids. And starting in the 90s, she taught herself to be a botanical expert. She took courses at the University of up in Oklahoma, Southeastern. Some of you may know, she was the great niece of Julian Reverchon, who was a collector in the Dallas area and lived in Dallas after they moved over here from France back in the 1850s. You know, she was a founding member of the Native Plant Society of Texas and attended all the symposiums up to her death. She's mentor to a number of people in the Dallas or in the North Texas area. Several of those are on the call today, particularly Mary Curry, and I know Jeff Quayle spent a lot of time with Mother, and Mary was instrumental in taking her out looking for wildflowers in her later years after she wasn't able to drive anymore. There's a nice article on my mother with in the Journal of Botanical Research Institute of Texas, and I think Tiana is going to post a link to that that'll give you a lot more detail about my mother and how people in North Texas thought of her mentorship. Some of her botanical activities, I was a little surprised to learn that she had as many collections, and TNI searched the TARCH database. There's 278 vouchers that she either collected personally or assisted in the collection of. And Britt has 156 of those vouchers, and 100 are down in the Texas Herbarium in Austin. You know, she collected primarily in Cook, Montague, Wise, and Denton counties, really in the North Texas area, where she became the expert of plants and flora in this area. She was kind of the go-to person. If somebody found something new, they always knew that my mother would be able to identify that. Most importantly, I think, to a number of people, is she kept a pretty detailed diary of each of the plants she found, when it was blooming, when it started to bloom, so you can kind of compare year over year what was going on there. Created a lot of plant lists for some of the special places that she spent botanizing. You know, she, traveled hundreds of miles each week, driving around Cook, Montague County, looking for plants, verifying that what she'd seen there last year was still there, etc. And she was a key one of the contributors to the Illustrated Flora, which is kind of the Bible for North Texas. I mentioned she loved to attend the symposium. She would even do some collecting on these symposium trips to different parts of the state. Attended some of the prairie conferences out of state as well. You know, she had left, when she passed away, she had 4 or 5 boxes of the old Sears J.C. Penney catalogs, which she used to press plants. So she would collect a plant, stick it in a paper towel, and stick it between the pages of the catalog. And I've been working with Tiana to be able to catalog those and put those into a spreadsheet to create a plant list from those. Next slide. One of the things that I'm kind of proud of is there was a new species that she discovered in Montague, and also I think in Cook County. And she was one of the first ones to document and collect a specimen of this Liatris atrophalis, commonly known as summer gay feather. And that species epitaph really means pertaining to the summer, so it's appropriate. You know, I think she had, you know, it wasn't until Guy Newsom and Bob O'Kinnon actually documented this species that it was given a new species name, but I think she was one of the first ones to collect it. And she has a number of plants of this genus that are in the herbarium details. Next. And one of the things that Mary Curry and Jean and others put together is a Shirley Day. This is a picture of the people that attended the 2018 Shirley Day on at the Thompson Foundation in Montague County. And that's a fun time to get together with fellow botanists and explore Thompson and other sites from there and have a picnic lunch. Next, and I'll turn it back to Tiana.
Thank you, Alan. I had the honor of meeting Shirley before she passed away, and I considered her a good friend. She never missed my birthday. I'd always get a card on my birthday. But I distinctly remember she knew the flora so well. And, you know, we have finite space in which to grow in the herbarium, and she had an understanding of the local flora to always be bringing us in big blotter papers with binder clips on all the edges to keep them flat and in good condition. And she would always bring us material of often species range extensions, something that looked strange, something that hadn't been recorded for the area. So you mentioned, Alan, that she contributed to the North Central Texas flora, and I certainly know she's acknowledged in those publications, our flora series. So thank you for presenting about Shirley, and something I hope that you may have taken home as a message from now is that we're reporting on what we know thus far, on what collections have been digitized that far. As Alan alluded to in his presentation, we're still working with all those Sears catalogs of specimens that Shirley collected. They never made her cut to be hand delivered to the Britt Herbarium for deposit, but they are vouchers of really incredible phenological information, and there are physical vouchers and good material because she knew what she needed to identify the plant. So they're reproductive. So Alan is helping us process that so we can bring those collections of hers to light as well. So it's my honor now to talk to you about Gaeta agilusc. So I think this is probably a name that many of you are familiar with, perhaps most so because of her publications, her popular publications, which you can see. I think these main 4 ones are down here on the bottom of the screen. We have the Wildflowers of the Big Thicket, East Texas and Western Louisiana, Butterfly Gardening for Texas, Butterfly Gardening for the South, and the one that I have on my bookshelf, which is the Wildflowers of Texas book. And on the back of the dust jacket of that book, you'll see an image of Gaeta with her camera. She's up there in the top right-hand corner. That's where that image comes from. So Gaeta was born in Mount Pleasant, Texas, which is in East Texas, east of DFW, in 1933. She married in 1949 and took on the married name of Amerson. So some of her earliest collections, you will see her as Peggy Amerson, and you'll see some of those labels in the next few slides. She was a botanist, a naturalist, a photographer, and an author. She supported and understood the understanding and implementation of native plants in daily life, and was really committed to communicating that to the public in a way that they could feel comfortable with and understand and own and use, not just for plants. She claimed the Cherokee as her people. Her name, Gaeta Agilesque, is of that origin. And from her book, Butterfly Gardening for Texas, she quotes, "Within our world, everything is of equal importance. Animals, rocks, plants, the waters, the sky above, and the earth below." She was a naturalist in that she was interested in all things and their interactions. And you can see that in her publications. Her books garnered various awards. I can't believe it's 2 of these books or their revisions that received awards 3 times given by the Native Plant Society of Texas, the Carol Abbott Memorial Award. And Gaeta, she collected plants for Donovan Carell for many years, and still the only treatment for all the plants of Texas was the Carell and Johnson Manual of the Vascular Plants of Texas. And Gaeta contributed and collected a lot of plants for that publication that enabled new state records, distribution extensions, things of that nature. She was a vascular plant field taxonomist for an environmental consulting firm, and she was very involved in the identification and study and mapping of rare and endangered species, and assisted with a lot of student projects. Part of this, or many of these, as during her time serving as herbarium botanist at the Tracy Herbarium at Texas A&M University. Or I suppose it was the biology department herbarium at that time. I'd like to also acknowledge that she passed away earlier this year. There is a wonderful tribute that was written about her by Dr. Will Godwin, who is pictured in the bottom right-hand corner of this slide. Will Godwin is the curator of the Sam Houston State University Natural History Collections. And that URL right there, the tiny URL, that is a link to the article, so you can find it. Again, I'll post this on the event page after today's event here. And I name Will Godwin because he was somebody that worked with Gaeta, that she trusted. And so she bequeathed her photographs, her slides, her archives, her field notebooks. Ashley was talking a bit about a collector and how they number their specimens. Well, a field log is something that's very important because it documents those collections in a notebook that they probably had out with them in the field. And so those are all deposited there at the Sam Houston State University Natural History Collections, and they can be accessed by contacting Will Godwin. If we look at her collections, if we look online in the Torch portal, which is something you'll likely hear about a little bit later if you stay with us to the transcription blitz, it's a way for you to access the digitized specimens for Texas and Oklahoma. There are over 5,000 specimens that she has collected, most of them in Texas, 140, 150 or so in Louisiana, few in Mississippi and Oklahoma. And the map on the left shows you the counties in Texas in which she collected the most. So the lighter the color, if it's white, she didn't collect in the county. If it's any color of purple, she did actually have specimens from that county. And you won't be able to read its name, but the county in which she collected the most was Titus County, which is up there in Northeast Texas. But you can certainly see her appreciation and interest in the biotic diversity of East Texas in this slide, and her specimens have really made it around. So they're deposited at 36 different herbaria around the world, and 81% of the specimens are actually at Britt and 13% at Texas A&M. She collected with various people, among them Barney Lipscomb, who's the director of the Britt Press and Library, Billy Helt, Dave Riskin, Francis Weir, and William Mahler. And Geraldine Watson and James Yantis are both 2 of the individuals with whom she collected the most. I think somewhere on the order of 200 specimens in each case, perhaps, but she most often reported having collected alone, being the only collector listed on that label. She did collaborate with Barney, Bill Mahler, and Eula Whitehouse at Southern Methodist University. And if you read Will Godwin's article, he talks about his excitement of going into her archives and uncovering more of a connection between her and Eula Whitehouse, who we haven't talked about today, but is another very important female collector for us here in Texas. And she is acknowledged, much like Shirley, with a lot of contributions for our Illustrated Floras of Texas program, where we're publishing floras for areas of Texas. We're currently currently in the process of working on our East Texas flora, and she's acknowledged in those publications. Here are some of her specimens for you to have a look at. You can see on the far left, this is a Gaillardia that she collected in Burleson County. This is with one of those collectors I mentioned, James Yanti, who got his PhD at Texas A&M. And you can see her name there in 1987 is Gaeta agilesk. And on the far right, you can see this hibiscus specimen was collected by Peggy Emerson. So this is rather interesting as women have gotten married and changed their names in some cases. It's a little bit more of a hunt to find and to credit them with their collections. So that's one thing we want to keep in mind as we're doing our investigations. Geraldine Watson was another botanist, another individual that she collected quite often with. And these are some of these specimens you're looking at here were actually cited, and she was acknowledged in various publications for her contributions to the understanding of the taxon of the publication. In particular, I'm thinking of hibiscus. There was a hibiscus publication that acknowledged her efforts for documenting the hibiscus in Texas, or in the southern United States. And then I did mention earlier that she had an interest in rare and endangered plants, so I couldn't help but get away with, had to put in a picture here of a sundew. So this is a sundew from Cass County, and one tiny little specimen on the sheet. I won't zoom into the label, but I've enlarged that single sundew so that you can see its leaves there at the base, that basal rosette, and you can see that inflorescence sticking up there. So please post questions for her and anyone else in the chat, and we'll answer them and talk about them afterwards. But it's my pleasure to introduce Natch.
Hi, I'm going to share 2 links in the chat. The first is a YouTube video, a short YouTube video from PBS about Yinez Enriqueta Julieta Mejia. And the second one I found actually kind of recently. I think it's more for young children, but I didn't realize it existed. It's a song about her, which I thought was fairly cute. Yinez Enriqueta Julieta Mejia is often considered the first Mexican-American botanist, and was notably one of the most prolific botanists of her time, which, given that she started at 55, her career is said to begin at 55 years old, and she worked for 13 years, is a fairly big accomplishment. So we'll talk a little bit about her life leading up to her career. She was born May 24th, 1870 in Washington, D.C. To parents Enrique Guillermo Antonio Mejia, who was a Mexican diplomat. And the reason why her and her family were in D.C. and her mother was Sarah R. Wilmer, part of a wealthy Baltimore family that included notable people such as like the 5th Catholic Bishop of Baltimore, Samuel Eccleston. A short while later, she moved to Texas, where she would stay for the majority or the early parts of her childhood. Mejia, Texas, specifically, which was founded actually by her grandfather, General Jose Antonio Mejia. And if if you're like me and you may not know where all the Texas cities are, I know we have some people from out of state. Mejia, Texas is actually just southeast of Dallas-Fort Worth. In Texas terms, that's not too far. That's surprisingly close. And it's just east of Waco. She spent her primary school years here, and the latter half of her childhood would be spent up north after her parents separated. She moved up to Philadelphia in 1879 with her sisters and mother. She actually had 6 half siblings. At this point in her life, while she was up there, she considered it very lonely. She was in a lot of boarding schools, a lot of Catholic schools. She moved in 1887 back down to Mexico to help her father tend to a ranch. And I think this is a pretty critical moment because originally she had intended to become a nun. And in coming down to see her father again, she was very excited to see her father again. She never got around to that. Although it is interesting, there are nun botanists that we have come across. In 1898, she got married, and he passed away only 6 years later. Her second marriage in 1908 was not as happy. It took a toll on her mental health, such that in 1909, at 39 years, she was advised by her doctor to move to California to see a doctor who specialized in women's health and who had founded a sanatorium where the patients recovered via occupational therapy and being outside in nature. So she would spend the rest of her life, approximately another 30 years, in this ecoregion. She fell in love with the San Francisco, California area and would stay there for the next 30 years approximately. So shortly thereafter, she actually divorces her second husband, takes back her maiden name, sells the ranch that he bankrupted, girl power. And in 1917 joins the Sierra Club, which had just been founded. She starts going on hikes. She joins the Redwood League, which also just starts up. This is around the time when we see the initiatives to start preserving land as national parks. She enrolls in UC Berkeley at 51 years old, 1921. One of her classes took her on an expedition, and it was that professor that kind of helped spark her interest in botany. So she starts taking botanical classes. Next slide. So she goes on to the California Academy of Sciences, which is to where we can owe a lot of the pictures that you see here. She was mentored by another prominent woman botanist, Alice Eastwood, who helped fund some of her trips. She was known for traveling solo. She has a couple of statements and quotes about that, which is pretty significant for the time period, and also even for now. She took trips down to Mexico and South America, and even up as far north as Alaska. It was in 1925, in one of her trips to Mexico, she came across one of the first new species that would be named after her. Mimosa mejia. So all totaled in her 13-year career, she wound up collecting over 145,000 specimens. Next slide. I have this quote here. I thought it was fairly a beautiful summary, not just from her, but also speaking as to herbariums in general, and this legacy that she leaves. I have a job where I produce something real and lasting. And on our left and on our right, we have some of the original specimens that she collected. On our left, we have Sororia Mejia, a type specimen. It's endemic to Ecuador and is listed in the IUCN Red List as endangered. Now, the interesting thing about a lot of these plants is that I can't find living pictures to show you what they look like. At least publicly on the internet, there are no live photographs. So what you see up above are two examples to help you get an idea for what that genus looks like. But over on our right is Mejianthus mexicanus, which is the only species in its genus. So I've left this part blank down here to kind of highlight the fact that there is no picture that I could show that either of the species or of its genus, it's just alone. And the IUCN doesn't have an evaluation for it either. It is native to Jalisco, Mexico. Next slide. This here is a specimen from the BRIT. It is, via the TORCH database, the only specimen of hers that we have at the BRIT of what is labeled as a Mundia regalis. So this is kind of where like we who are looking at like these specimens from like our past, and we kind of come in and have a little bit of a journey across the generations, where she labeled it as Osmundia regalis. And I have some pictures over on the right of Osmundia regalis from iNaturalist. Next slide. But as we can see, European royal fern is, well, only native to Europe. So it was actually kind of interesting, buzzing around, figuring out, well, wait, what happened? Well, since she collected it in the time since she's collected it, it has actually been that Osmunda spectabilis, which used to be considered a variety of regalis, is actually considered its own species. And as you can see, it's actually found in the West where she found it. And it's just been an interesting journey there. So specimens are basically constantly being updated. And this is I think, a neat segue into our ending.
Thank you, Natch. So I hope what you've heard from each of our speakers today, and thank you, speakers, to each of you, and also thank you to the audience who's here today that's allowed us this opportunity to talk about some of these women that you may or not have encountered before in your own explorations of botany and herbaria. But hopefully we've impressed upon you that collections are of vast import as we face our future. You're looking at an article from 2004 talking about the decline of plant collecting in the United States, and that this is truly a threat to the infrastructure of biodiversity studies. One of the tenets of our proposal that was funded to digitize our specimens here in Texas and Oklahoma was that we're in a unique space where we are at the confluence of so many different climatic variables, of changes in temperature and humidity and moisture, regimes. And many species find the eastern extent of their distributions, the western or northern extent of their distributions in our area. So these specimens represent very important elements as we look at these studies to know how can we mitigate, how will these species react as climate changes take place. So collections become even more important, and it's a way for all these women to live beyond their time and for these specimens to be used beyond their time. One of the reasons we really plan to use Bionomia to credit these women with their collections is because there are further ways to acknowledge the use of those specimens in current and recent studies. So I encourage you to explore that website. It's bionomia.net. Another link we'll post to the event page. But just a reminder before everyone heads out and starts collecting, these are some of the best practices that we like to share when we give our presentations on collecting. And those are that you should always collect with permission, with purpose, with a conservation ethic in mind, good, properly preserved specimens, and deposit them in a publicly accessible herbarium. And to end on another note, you're looking at this slide with photographs of the individuals that identify as women, some of them that are as research associates or staff currently here in the Botanical Research Institute of Texas at the Fort Worth Botanic Garden. You're looking at left to right, the top row, Ashley Bordelon, myself, Brooke Byerly Best, Kimberly Sasson, Rachel Carmichael, and then left to right on the bottom row, Erin Flinchbaugh, Jessica Lane, Alejandra Vasco, Megan O'Connell, and Manuela Del Forno. So these are some of the collectors that, at least out of our herbarium, are continuing to deposit in these collections. And so we'll do our best to make sure we are putting ourselves out there as well, both in support of other women in science in general, in collecting in herbaria and botany specifically. And of course, we're always excited to answer any questions. And there were a few that were posted in the chat, but if there's anything that has come up, then please feel free to add it to the chat now. And I'll ask all our presenters if you could share your videos and be prepared to unmute. I am going to navigate back to one of our earliest questions, which was really interesting. And because Sue is not here, who did the research into Agnes Chase, I'm not sure that we can necessarily address it, but Kimberly, in your preparation for the talk, did you discover anything about how Agnes Chase was able to support her own research?
I think I might be able to guess at some of it. I think a lot of her suffragette experiences led her to be in communication with a lot of different people from a variety of backgrounds. So I don't think she personally funded it as much as she raised funds from not only the civil liberties type organizations, but also from the people that were botanists in other countries. I know when she would travel, she would interact with a lot of other botanists there, and particularly women that were interested in botany, and they would room together and things like that. So my guess is that it came from her contacts, her wide, extensive contact.
Thank you, Kimberly. And Natch, if I recall, you were talking about Ines Mejia and the fact that Alice Eastwood had, will you tell us?
Yeah. Alice Eastwood helped sponsor some of her trips. In particular, I was able to find details on one where she met another woman, Hopkins, Hopkins, wait, no. I am so sorry. I wrote so small. Roxana Stinchfield Ferris, thank you, at Hopkins Marine Station. Yeah, and she took a trip with her and her husband and asked for funding from Alice Eastwood, who granted it under the condition that she be paid per specimen that she collected. So.
Thank you, Natch. I imagine, yeah, thinking about today and how women are raising funds for their work today, you saw on the previous screen, Alejandra Vasco, who she is one of our research botanists here, our fern curator, and she's currently in Colombia on her Ferns of Colombia grant, funding she and collaborators, other PIs raised to do work on the fern and lycophyte flora of the country. And in fact, I think right now we're prepping for a trip to the Philippines. Manuela del Forno, who was also on our previous screen, not plants, but lichens, which we also store here in our herbarium, but Manu is on a grant, another National Science Foundation grant to fund a collection of lichens in the southern Philippines. So the National Science Foundation and many other private foundations and other organizations help fund this kind of research. The Native Plant Society of Texas has done so in the past, the Garden Clubs of America, and there's many others I'm not mentioning right now. But certainly it's a hunt for that kind of funding. Are there any other questions anybody would like to ask in the chat? All right. Well, please feel free, herbarium@brit.org. One of us will put that in the chat to make sure you can contact us after this. Thank you to all of you for your attendance today, for your questions, your comments. There were some other comments in the chat as well that we'll be saving. We would like to hear from you, so do tell us if there are other botanists you'd like to know more about or that you'd like to tell us about, because we'd love to learn and hear from you, and we recognize that it takes a whole lot of us to unravel some of these stories. We'd also like to thank our volunteers. All our volunteers, many of whom are on this call, especially our armchair botanists here at Brit, because it is the efforts of these volunteers and staff at other institutions and volunteers at other institutions digitizing these specimens that brings them to light. The Texas-Oklahoma Regional Consortium of Herbaria and all our collaborators in our two-state region and beyond and in the wider herbarium community, National Science Foundation for the funding of the grant to digitize Texas and Oklahoma specimens. And then this is a thank you for the next portion of our talk, which is a thank you to Mike Denslow from Notes from Nature, who has helped us put some of our specimen images online so that volunteers and staff can digitize, can transcribe information from those specimens. And I'll go to the next slide and hand it off to Ashley, who will take control of the screen. So thank you for attending, and we will now head into the transcription, the guided transcription blitz portion of our presentation. So if you've got to go, thank you so much for attending. And if you'd like to stay, Ashley, take it away.
Yeah, so for those of you who are interested in actually getting some viewing time of some of these specimens that we've kind of briefly looked at, this is a way that you can help us by digitizing these collections, and you will definitely come across some of these collectors that we've mentioned in this project. So where we are now, this screen that you're seeing is a platform called Notes from Nature. And it's a way for institutions, not just plants, but others to create projects that gets community scientists involved and volunteers, helping with different types of digitization from measuring to entering data on all sorts of things. So once you go to this website, the first thing that you'll want to do is either create a username, and if you already have one, sign in. You don't need a username to use the platform, but it's a great way to save any cool images that you've run across and ask us questions directly about anything that you have. Typically, if you're on the home page, you'll actually see our project under active projects. So it's this torch, the Texas and Oklahoma image right here. So I'm just going to pop into that. And typically, if this is your first time popping into one of our expeditions, so this is just the main project page. And then we have an expedition right here under Get Started. So it's our 29th expedition. Right before COVID, I believe before COVID, we had started a few of these, but this was a great way for us to continue working, even though we were remote, and to continue our responsibilities for our grant with digitization. So the cool thing about this project page is you can go to the statistics and you can take a look and see kind of the progress of each of the expeditions. So this 29th expedition only has a few days left. So there's not too many more images that need transcribing. And then we have 2 more in the docket ready to launch once we've completed. And let me go back. You can always click Learn More here. And this just gives you a little bit of a background about the project, as well as a general FAQ. But I'm going to walk you through step by step, but this is just a little bit more instructions on specifics, some typical things that we get questions about. So it's a great tool to look at before you get started. But let's go into Expedition 29. And typically, if it's their first time going into this expedition, you'll get a window that pops up asking you to scroll through the general instructions. So that's great to look through, but I'm going to go through that right now with you. So here we are. We're in the expedition, and you can see we have an image of an herbarium specimen on the left, and we have some fields to enter in data on the right, our tasks to do. So the first step is identifying where we need to look for data on this specimen. So everything we want is in this label in the bottom right-hand corner. Sometimes you'll see it in the bottom left, but it's typically in the bottom right-hand corner. There's a few controls to zoom around the image. So I'm going to zoom in. I'm going to click and drag. So this label is right next to the fields that I want to enter into. So typically the country isn't going to be listed if there's a state, it's just kind of assumed. These in the expedition should be Texas, but we have found a few other states have been sprinkled in. So you may come across a few other states. The great thing about this locality is that there's a drop-down box available. So as you type the county, you'll see it pop up. So you don't have to worry about any misspellings. Under each task, as you go through, you'll see a button that says need help, some help with this task. If you click it open, it'll help you identify what fields you need to be looking for on the label, and any other information about those fields can be found in that help button. I'm going to click next. And the next prompt is the scientific name. So the scientific name is going to consist of a genus and a specific epithet, potentially a variety. So in this field, we don't need a family name unless that's all that's given, and we won't include the authorship. So in this example, on the left-hand side, the scientific name is italicized. It's Galactia volubilis. So I'm going to type that in right here. And like I said, we don't need the authorship. So the L. Britton at the end is basically just telling us which people helped describe this species. So the L is for Linnaeus. You'll probably see L a lot after some scientific names, and then also Britton. So afterwards, Britton came along. So collected by this example, we have a few names down here, but it looks like the first collector is Evan J. Basha. So I'm going to start typing that name in. And if I click need some help with this task, you'll see that it says the name of the person that collected the specimen is who we want in there. But if there are multiple collectors, then separate the names with a comma. So this is that example. We got a whole list of people who went out to collect on this day. So I'm not going to include the word with, and I'm just going to start typing the other names that follow. Feel free to let me know if I type anything in wrong.
Ashley Janice's name is spelled incorrectly. Ashley has perfected
Here we go.
the skill of talking and typing precisely at the same time.
And it looks like Evan's number is 104. So that collection number will typically come after that first collector. You may see other numbers written around the sheet. So just look around the label and see which one makes the most sense to be that collector's number. And it's typically going to be the one that follows directly after that first collector. And then lucky for us, we just put in the verbatim date as it's on the label. These all look good, so I'm going to click next, and this is the final task. Basically, the rest of the information on the label that remains. So we don't need to put Texas or Guadalupe County because we've already included that in the primary locality in the beginning. So what I'm going to start off with typing is what follows county. So collected near Mill Creek, along Mill Creek Road. We have some coordinates in here. If someone can help me remember it's Alt 17. I know Alan knows it.
So you'll see looking at this label that they can really vary what people choose to put in and on their labels. And so kind of a study of these individuals' labels give us a little bit of insight into them as a scientist, as a person.
Yeah, this label makes me think this might be a student collection, potentially with a bunch of other people. Oh, and the great thing about this primary label text field is you can expand it if you need to be able to see everything that you're typing.
Actually, Ashley, you're right. So Alan Livens would have been the curator, I think, at the point when this was collected at Texas Lutheran University Herbarium.
Station 168. Purple flowers. So we got some description there. And that is about it. So at this point in time, if I think that maybe I forgot something or maybe had typed something in wrong, I still have the option to go back. And it looks like everything is still saved that I typed in. So we have two options at this point. If I felt like something was off about this label, maybe the label was cut off and I couldn't read some of the information on it. Or, it could be a number of things that may have been confusing. Each specimen is its own case. So there are a few tricky ones. So clicking the Done and Talk option, which I'll go to now, will bring me to this page right here. If I scroll to the bottom, I can leave a note about this image. So this is that place where you can leave a question if you have it and be as specific as you want. And that way we can get back to you. And the cool thing about this is you can use hashtags to specifically tag that your, whatever is of question on the label. So if it was the collector, or maybe it was the scientific name, illegible. So those are some ways that we're able to use, we're able to find certain issues. Linda in the room. So you can leave the comment here, and then going back to the project, we can get back into that expedition. Let's say you come across a specimen and the label, you can't even put any information at all. It is.
Look at that label, Ashley. Look at
we got a Peggy Emerson label.
Yeah, Gaida's collections.
Lucky us. So you do have the option to not transcribe a specimen. If it's too long, I know some people see a huge label with a bunch of information and they just don't want to deal with that. And that's totally fine. If you click the refresh button, you'll get a brand new image, which is great. So in case you really didn't feel like you were able to put in any information for that label, you can always get a new image by clicking refresh. You'll see some of these other options at the bottom. These are the cool things if you have a sign-in as well. So if you have a login, you can leave a done and talk message. If you aren't signed in, you can't. If you're signed in, you can favorite images. This is a good example, actually. The handwriting or the typewriting is a little bit faint. So there's an option to invert the image. This may help you read some handwriting. Maybe it didn't help us in this case, but this is also an option to help you transcribe. Some of the other options, if you click the I button, you can specifically go to, oh, I clicked the wrong thing. That was taking us to iDigBio. There is usually an image button. I'm not sure why I'm finding it right now. And then you can also create a whole category of images that you want to save. So for me, I have historic Texas botanists and NLU collection stories. So interesting ones from our NLU expedition that I've tagged in there. Okay. So let me keep walking through this one, this example. And as I do, please let me know if you guys have any questions so far. But the great thing about Notes from Nature is just how user-friendly it is, which we love. So more people are able to help us out, even if you don't have any botanical experience. We never want people to think that they have to be an expert to help us out.
It's interesting as we transcribe specimens, you know, it's very important to do a verbatim transcription to the best of your ability, but there are some things that you have to interpret to some point, which not yet at this point, but if you see the date on here, it makes you wonder, was that collected in 1952, 18, you know, so there's all sorts of clues you can use about the specimens and the types of paper and the other context of the label that can help you know what year it was collected. So a lot of this work that is happening right now is actually done by volunteers. So a lot of the specimens, perhaps most of the specimens that you saw today in our presentation were transcribed by volunteers and then brought in by staff to our portal online, which perhaps we may conclude this presentation with at closer to the half-hour mark, is to show you how you can access the data that have been digitized thus far.
Okay. So in this one, I just clicked done. And once I clicked done, that image went away and it automatically brought me up a new specimen to transcribe. During our weekly, well, used to be during our Armchair Botany sessions, when we answer questions from volunteers and go over some cool plant facts, some of the things we like to do is guess the plant. So what we'll do is just being able to look at the top of this image and not the label, seeing if any of us know exactly what the identification is. So this could help you with your botany skills.
Oh, look, don't look yet. Oh, I was gonna have folks guess.
You can do the next one. Yeah, I would have maybe guessed APAC for this one. Another Lutheran University. Some of these same collectors. So I think these are definitely some student collections, which is also so great. I know that we have collections of students that were students of some of the folks that we've actually talked about. I know Connie Taylor had a few students under her that we have collections from, not only in the Britt collection, but also our NLU collection in Louisiana as well. And I wonder sometimes if people, past students, think where their collections are. And so hopefully as we digitize these, maybe they can discover them one of these days. Elise.
One thing I'm not sure we've talked about too much is that Ashley communicated the idea of a collector name and number, Amy Lee's collection number 37, but I'm not sure we really discussed the idea of duplicate specimens. So Amy may have collected 5 specimens of Tortoise arvensis from the exact same population on the same date and time, and those 5 would have gotten identical labels. They all would have been Amy Lee's number 37. The idea being that they can be sent around like trading cards to other herbaria. It's an insurance policy because that collection exists in multiple places. So something happens to one herbarium, those specimens exist elsewhere. It's also a way to increase access to those specimens. So one of the benefits of us digitizing some of this material is when we put it online in our portal for everyone to access, hopefully those data are discoverable by other institutions that may be digitizing their specimens. A lot of the specimens we're coming across were pre-digital or were typed up and never saved as a database. They were put into a Word program that was then lost. Ooh, any ideas as to this specimen?
I'm thinking it's a monocot, maybe.
Parallel veins probably on the leaves if we were to zoom in, long grass-like.
And Tiana, you mentioning duplicates, I mean, even is so relevant even today when, you know, in recent history, herbarium collections have been lost in various ways, whether it be fire, flood. So it really is important that these collectors do collect in duplicate and have, you know, them elsewhere in case they are destroyed in those other places, that we still have documentation and proof that these plants existed.
Ashley, we have a suggestion from
Right.
the audience, and my bet is on her guess. So Lisa suggests it might be a blue-eyed grass, Cicericium.
Sure is. That was a good guess.
Not only is it Cicericium, but you're seeing a name there that Alan talked to us about today. It's like you planted this specimen, Ashley.
I know. Yes, this is a Reverchon collection, and especially with some of these really old collections, I'll zoom out so you can kind of see, you see multiple labels. They are duplicated information, but you can see that, you know, Missouri Botanic Garden had this specimen at one point, maybe used his field logs to create a label. And then another label was also created with some of the additions from the field label. So there's a lot of data on here, but they are the same. So you may see this with some of our older collections like Reverchon and Lindheimer. Because there's so little data, it's been pulled from a few places, so we have the most that we can.
There's a good question from Mary Curry, who also worked with Shirley Lusk. Mary, I'm glad you're here with us today. And Mary's asking why we don't include the original herbarium where the specimen was originally deposited. So that's actually a tricky question, because if you look at that specimen that Ashley was just mentioning, that specimen existed at one point at the Missouri Botanical Garden. And if you scroll up to the top of the image, you'll see a different stamp in there. So Southern Methodist University. So that kind of accession history as far as who owned the specimen or who distributed that specimen can be important, but it's not something that we currently record on our specimens. And it's mostly in the interest of time because we feel that the most important needs to access our specimens are the species names, the locations, the dates for the sorts of research we're doing now. And I think something we've all learned along the way is that the digitization of these specimens, the databasing of these specimens is a work in progress. This record is still not complete. It will evolve, it will improve. It will improve even further when we do what has been done handwritten on this specimen, when we are able to tie the field notes of these collectors, like Gaida's field notes, to some of her specimens, and we can uncover more about that collection event in the future. So it's not recorded right now. It doesn't mean it's not important. It just means it's not been a priority for us at this time.
Well, and I would just add that Julian Reverchon, a lot of his collections went to Missouri Botanical Garden originally because he was collecting for Asa Gray and some of the other botanists of the time.
That's such a good point, Alan. If you look at our proposal, it includes mostly herbaria in our 2-state region, but it includes other herbaria, Kansas universities and the Missouri Botanical Garden and New York and some other institutions that do have some of our historical collections because there weren't herbaria in Texas to house them initially, or the collectors that were collecting, exactly as you said, Alan, were collecting for those other herbaria. We're at 1:24, and so we'll conclude our session at 1:30. Please go ahead and ask any other questions you might have in the chat. And then I think perhaps when Ashley's done with this, we may pop quickly into the Torch portal, which is where these records will end up after they go through a few more steps of data cleaning. And I'll share that. Oh. Someone's ahead of me.
You do it.
Thank you, Ashley.
Yeah, so this is the place for you guys to search specifically our collections, but really any collections from any herbaria that you'd like. So if I go to the search button at the top and search collections, it'll bring me to this page right here. And if you look, it says select, deselect all collections. So right now, not only are all of the Torch herbaria selected, but also from all these other herbaria across the United States. So if we want to specifically just look at the Britt collection or the collections with us, I'm going to deselect everything and let's just search our collection right here. Once you get to this page right here, this is all the search criteria. So maybe you're interested in just a specific county that you live in, or maybe you're like us and you're starting to get interested in specifically collectors and where they collected. This is the spot. There's so many different options for you to search. So I guess I'll do Texas, Tarrant County. And I won't include any specific collector's name, and maybe I'll just limit it to specimens with images. You can also choose specimens that have coordinates, so you can see exactly where they were collected, or at least an approximation. There's 2 different display options. You can do either. They both serve kind of a different purpose. So I'm going to select the list view first. In this view, it's only 100 records shown at first, but it's great because you can kind of get a small preview of some of these images on the right-hand side. But you'll see some of that label data already appearing on this page. And yeah, from here you can go into the full record. I guess this is a good example right now. It just popped up for this cactus species. Locality details protected, protection typically due to rare or threatened status. So current user has been granted all status. So I'm able to view this because I'm logged in, but there may be some specimens that you can't see the locality for that exact reason. So there are some protections on certain species in searches. But let me click into the table view. So you can look at this, and this gives you a little bit more of a search functionality where you can sort it a little bit better. So we're in Tarrant County, so maybe I want to sort it by date, or maybe a collector name first and then date. So let's do that. And it might take a little bit. Oh, that was pretty quick. So we have some folks who don't have a number. We have Albert Ruth, who was one of the early botanists in Tarrant County. So we have a lot of his older collections in the early 1900s. Yeah, and so from here, you can see on the left-hand side, there's a little camera icon. So that'll tell you if you didn't select specifically only specimens with images. If you just did a generic search, you would know from this icon that you'd be able to see an image in there. And let's say that you see something that's completely wrong. You can submit a comment to us. Or if you're an expert on a certain taxa and you think something is identified wrong, you can also put your comment there for us to update things. So just another great way to get involved with our collections by searching around and helping us out. So eventually those expeditions and notes from nature and all of those images and the data that the volunteers help with us will be going back into the Torch portal. So if you do see something interesting in one of those expeditions, know that there's kind of some time in between where it'll actually be searchable for you in the Torch portal. Is there anything I'm missing, Tiana?
No, I think that's wonderful. Hopefully, actually Lisa alluded to something that that this is one of the specimens you looked at was a good example of the handwritten scientific name being hard to read. So starting with the beginning of the pandemic, we went to almost weekly armchair botany sessions, which was a way for us to stay in touch with our volunteers and the people that wanted to continue to engage with the specimens. We have since moved that to approximately once a month. The schedule for those armchair botany sessions will be posted on the website I shared a little bit earlier, which I'll share again. I'll share again in the chat, or maybe Ashley again as I'm talking. So we invite you to come and join us. Those are very informal discussions. We'll be moving forward with a model where we have short presentation, about 30 minutes perhaps, and then we move to just a discussion, because I think there are many people that are on this Zoom session right now that have participated in that. And I think they might all agree that we all learn quite a bit from one another and get to talk about quite a variety of topics, not just plant identification, but collectors and history.
Yeah, so if anything was interesting today, it is very similar for our normal armchair botany meetings, which we enjoy very much. Like Tiana said, not only do we hopefully teach you guys some things, but we learn something from our guests every single time. So it is such a fun time for us as well.
So thank you all for attending, and please don't hesitate to email us or contact us in any other way. You can also find us on the website. Thank you so much.
Thank you, everyone.
Goodbye.